Hi everyone, I'm Yu-Hsiang and from UC San Diego. Today I'm going to present our work, Ultrafast and Ultralarge multiple sequence alignment using TWILIGHT. Here's the outline for today. First, let me introduce multiple sequence alignment. Multiple sequence alignment is to align homologous characters across multiple sequences. And the goal is to reveal similarities and evolutionary relationships between multiple sequences. An example is shown here. So the alignment can be easily achieved by inserting gaps to some of the sequences and make it look like the same character looks like they're aligned together. So multiple sequence alignment is a cornerstone of many bioinformatics applications. For example, it can be served as a training data for machine learning models, such as AlphaFold to predict protein structure. It can also use to construct pangenomes and use to infer evolutionary trees, to identify conserved region across multiple species, and it can also be used to reconstruct ancestral sequences. So there's a few limitations of the existing MSA methods. So a previous work by Simonov shows that the fastest tool up here requires 77 hours to align 1 million sequences. And the sequences here are short. They're only average 1,500 bases. And also, none of these existing MSA tools can handle tall and wide alignment. So tall and wide is based on how we present multiple sequence alignment. Tall means many sequences and wide means long sequences. So as you can see here, apparently the fastest tool MAPT here, it does not scale linearly with sequence length. When the sequence length increased by 10-fold, the runtime increased by almost 40-fold. And this poor scalability to sequence length make long sequence alignment infeasible. In addition, current MSS tool does not have very good efficiency on modern HPC platform, as you can see here. The best tool here, Pasta, saturated at 64 cores, and other widely used tools like Math and Tcoffee does not really scale well over 8 CPU cores. And to date, there's also no widely usable GPU accelerated MSA tool have been developed. So why do we need a faster and MSA tool that can use for long sequence? So there are many applications. I'll just name one here. During pandemic, there are millions of SARS-CoV-2 sequences has been sequenced and sampled. And there's a need to align all those sequences for various applications. However, as I mentioned before, none of these MSA tools can handle these scales of alignment. So some MSA tools like Viral MSA, WMSA2, and H-Align3 apply some reference-based or center sequence-based heuristic to speed up the process using pairwise alignment. So it might be useful for some cases, but they have very limited applications. And since they're aligned to reference sequences, they might have suffered from reference bias, or they might lose of phylogenetic information. So now let me present you our work, TWILIGHT. So TWILIGHT stands for tall and wide alignment at high throughput. So there's two modes in TWILIGHT. First, the default mode, just like all the multiple sequence alignment tool, we took in a guide tree and a row sequence file as input and generate the outputs as the multiple sequence alignment. However, since the guide tree is not always available, we also provide TWILIGHT iterative mode that use external guide tree inference tool to estimate the guide tree and follow the same procedure as default mode to generate multiple sequence alignment. And also, iterative refinement is also like, it can also be used in our TWILIGHT iterative mode. Let me show the comparison between TWILIGHT and existing MSA tool. So MAPT, Lindsay here, have the highest accuracy, but it barely scales over 200 sequences. And Migas and Pasta are MS tool that builds upon MAPT Lindsay with their own unique merger strategy. They have high accuracy, and also they can scale to 1 million sequences, but you just need a long enough time for them. And the fastest tool, MAPT here, the fastest option of MAPT can do a tall and wide alignment, but they might suffer from a lower accuracy. In our work, TWILIGHT not only can generate a taller and wider alignment than MAPT, but also in a shorter time, but also have a higher accuracy, like comparable to Megas and Pasta. So the figure shows an example of TWILIGHT alignment. Let me present the result of TWILIGHT. First, TWILIGHT successfully generates a multiple sequence alignment of 8 million SARS-CoV-2 genomes. and it only needs around 30 hours. So to evaluate this alignment, since the alignment is really large, we compress it using PenMan, which is also an accepted talk in ISMB this year. So people who are interested in may refer to a recording of July 21st under the track of GenCom Bio. So first, to evaluate the alignment quality, we first compare our number substitution to Archer match. So we achieve a high concordance to Archer match of 94%. And since Archer match does not have indels in it, we show our results by this table here. We compare to all the previous studies, and we find that our MSA accurately capture the lineage-specific indels reported in prior studies. Now let's go to simulated data sets where we have a golden reference alignment. So as shown here, our RNA-Sim data set is a challenging data set that have at most 1 million sequences, and it often used to evaluate the quality, the scalability and the accuracy of MSA tool. As shown here, TWILIGHT achieved the lowest error rates compared to other state-of-the-art MSA tool. Now let's go to the scalability and the speed of TWILIGHT. So just a reminder, the previous work shows that the fastest tool needs 77 hours to align 1 million sequences. And in TWILIGHT, we only need 32 minutes to align 1 million sequences. And this is for the CPU version, so which can be further accelerated by adding GPUs and achieve a 13 minutes of performance to align 1 million sequences. Also, as shown in the figure here, it's obvious that TWILIGHT scales linearly with sequence length and make it possible to align very long sequences. As shown in the result here, we can align 10K sequences with 1 million bases in under 4 hours. And thus, TWILIGHT can, as shown in the figure here, since our cluster can only have 128 cores, but you can see that there's no saturation now until now, so we can expect that if we have a cluster that have more than 128 cores, we can even achieve more speed up with our TWILIGHT. So TWILIGHT is a great option for modern HPC platform, but it can also be used on our personal devices. So with a divide and conquer strategy, which I'll mention later, we can successfully reduce our peak memory consumption of aligning 1 million sequences from 133 gigabytes to under 8 gigabytes. That means that it is also possible to align 1 million sequences, that's large-scale alignment on our personal laptop. And this result shows the efficiency of our iterative mode. As you can see here, TWILIGHT iterative mode can lower the error rates of alignments and trees through multiple iterations. The NRF here stands for normalized Robinson fault distance. It's often used for compare the difference between two phylogenetic trees. And lastly, TWILIGHT is originally tailored for align long nucleotide sequences, but in the latest version of TWILIGHT, we also add supports to perform alignment on protein sequences. However, our speed is decent. We are 2 times faster than MAPT, but as for the accuracy, there's still a gap that we need to fill. And this is definitely ongoing process that we'll try to fix it in the near future. Now let's go to the method of TWILIGHT. So TWILIGHT is based on multiple previous progressive alignment algorithm. We use a progressive alignment, and we also represent alignment using profile. A profile in our definition is a combination of character frequency of each columns. And we also adopt a needle-moving algorithm, which is a global alignment algorithm with a fine gap penalty, and also adopts the position-specific gap penalty introduced by Julie Thompson. So the high-level idea of position-specific gap penalty is to give a lower score to columns that already have gaps in it to make it easier to create a gap there. So here's a few challenges on toad and wide alignment. First, high memory usage when storing large data sets. For example, we need 24 gigabytes of memory just to store a row sequence of 8 SARS-CoV-2 sequences. So we present a divide and conquer strategy. We first divide a large guide tree into multiple subtrees with leaves no more than the maximum subtree size. And we do the alignment sequentially of all these alignment, and then we merge it at the end. So when merging these alignments, we represent it using a profiles and binary string. So for more details, please refer to our publications, since I don't have much time here. And another challenge is to is like there's significant memory required to store traceback pointers for long sequence alignment. So here we introduce a few alignment algorithms. So the traditional full matrix algorithm is not practical because both time and space scales quadratically, and Bendy alignment tries to solve this issue, but the space complexity is still not practical. So tiling algorithm is introduced that use only constant memory to store traceback pointer, but it has a lower accuracy. So we use a recently introduced algorithm called telco algorithm. The high-level idea of this telco algorithm is that it can use constant memory, but also achieve optimal alignment under Bendy constraints. And the less challenging on tall and wide alignment is the scalability limitation. So as you might imagine that when a gap is inserted in the alignment, they'll always be there. And as we proceed to a later step in the progressive alignment, the alignment length will be longer and longer. And since we know that the algorithm scales linearly with the length, and it might become slower and slower. So we remove gappy column before the alignment, and we add them back after the alignment to minimize length expansion in the alignment step to keep our alignment step faster during the whole progressive alignment stage. And a result shows here that our strategy not only improve the speed, but also lower the error rate. Result is shown in the table. Okay. So, and TWILIGHT also, we try to also explore all the possible parallelism on both CPU and GPU. So GPU version of TWILIGHT, the pipeline is shown here. CPU will compute the profile, which turns the MSA into profiles, and will send the profile to GPU. And then GPU will do the alignment step, and then send back the traceback test to CPU. So as you can see here, all the steps, if we can find any independency in it, then we will try to apply a parallelism on it. Okay. And here's the way that we parallelize the alignment step on GPU. So there's a 3 level of parallelism on GPU. First is the intra-alignment parallelism. So following the progressive alignment, we can find that there are many alignment pairs that are independent to each other. As you can see, since they are independent, so we can apply parallelism on it. We assign one GPU block to one alignment pair. And within each alignment pair, following the dynamic programming, the calculation of score of each cell is independent when they lie in the same waveforms, or we say same anti-diagonal. So we can assign one GPU thread to one cell on the same wavefront. And in TWILIGHT, we also support multi-GPU parallelism. So we have tried on at least 4 GPU parallelism, at least 4 GPU, and it still works. Okay. So here, it comes to the conclusion and future work. So we present TWILIGHT and MS tool that overcome the scalability limitation of existing MSA tools. So we maintain a constant memory footprint using telco algorithm. We present the slowdown by removing gappy columns before the alignment step. And we also efficiently leverage the available parallelism of modern HiTSeq platform which contains multiple CPU cores and sometimes contains GPU cores. And we also significantly reduce the memory usage by our divide and conquer techniques. So let me reinforce our results. TWILIGHT can align 1 million RNC sequences in 32 minutes with CPU version and with the lowest error rates compared to other tools. And we can align 10K sequences of 1 million bases in just 3.35 hours. And to the best of our knowledge, TWILIGHT is the only tool now that can perform a reference-free MSA of 8 million SARS-CoV-2 genomes. As for the future work, we try to incorporate the more sensitive method for highly divergent alignments. And also, just as mentioned before, we'll try to improve the accuracy on protein alignments. And we plan to expand TWILIGHT to a multiple whole genome aligner. So there's plenty of multiple whole genome aligner like progressive characters, pangraph. So they are useful, but their speed are still too slow. So with the power of TWILIGHT, we're planning to extend TWILIGHT to do whole genome alignment and also achieve a speed up to all the prior works. So for the acknowledgement, I would like to thank Smith and my advisor, Professor Yatish Turakia, and all the people listed here that have helped us during our development. And TWILIGHT now is available on multiple hardware platforms, and can be installed through Bioconda, Docker, and of course, you can build TWILIGHT from source. And last but not least, I know there's a lot of things here, but thank you all for your attention. Thank you.
Thank you. Are there questions? Okay, so there, number 2.
Hello. Thank you very much for the presentation. Very impressive results. I had a question about the removal of the gappy columns.
Yes.
You put them back afterwards. This seems very smart, but do you have like theoretical guarantees that this still produces the same alignment as if you didn't remove them? I mean, you take them out, then you align, then you put them back in.
So yeah, let me try to. We remove the gappy column as shown in the figure here, and then we perform the alignment, which get us the traceback path like this, like N mismatch or mismatch and I misinsertions, and we just simply add the gappy column back to the position that it used to be. And if 2 gappy columns are added to the same position, we just simply merge them. And since gappy column contains most of the gaps and they do not contain much information, so in this approach, like merging 2 gappy columns does not have much effect on the overall accuracy, but it can prevent to generate a very excessive large alignment that we might observe in pasta or megas.
And you don't have a problem where, since you took out a gappy column,
yes,
you might have problem aligning the bases around that may not have the same alignment as if the gappy column was still there?
Yeah, it is also possible, but from our experiment, like with this approach, we actually improve the accuracy, because I think gappy columns sometimes may cause many issues in the alignment, because the gap will serve as like a penalty. And if it's a gappy column, which means that if you follow the idea of progressive alignment, which means that it might not have much, like his children or like his parents did not align to this column, and we might I think I would say that it's reasonable to exclude it before the alignment. Do I answer your question?
Yeah, it makes sense. Thank you.
Okay, another question on number 2.
Yeah, this is really cool work. I think I haven't seen many MSA programs for GPUs, so this is great. I do have a question. What are sort of the limitations on identity that are needed for this to work well? Does it break down at some point if the identity of the sequences? I saw you did proteins where identity is really low, but like,
I see. So
where's the protein threshold or the identity threshold here?
So I would say, like, for protein alignments, since there are 20 characters and in nucleotide there's only 4 characters, so it's easier to align nucleotide sequences than protein sequences. And since now we only simply replace our like scoring metrics from nucleotide to protein, and we haven't done our optimization on protein alignment. And I believe that, for example, there's a talk in ISMB this year, like FAMSAT2, that did very, very good work on protein alignment, and they do have some techniques to just to optimize for protein alignment. And I think there might be the like difference between like nucleotide and protein alignment of our result. And as for the sequence identity, I'm not sure if we can directly compare like protein to nucleotide, but we do observe that for like nucleotide sequences, if the sequence identity is lower, like our RNA sequence is already a lower, like low sequence, but if the sequence identity is too low, then I think none of the MSA tool can perform well on it.
I guess, so you don't know what, like, if it's 80% that you need or 60%?
No, I'm sorry, I don't know if there's a threshold that causing this issue.
Okay, cool, thanks.
Okay, thank you so much. Let's thank the speaker again.